everyone, and welcome. This is Professor Hall, and today we're going to be talking about college success reading and note-taking strategies. Hopefully, these are some skills that I'm giving you pretty early on. I'm going to be explaining things, and then you'll be doing some practice and some discussion. But the nice thing is that this is not an exercise that is kind of in a vacuum or a bubble, right? Meaning it's not just for this class. These are things that I'm hoping, as the title of this presentation states, that you will be able to use in your other courses and possibly in your future careers. So let's get started. So, reading and note-taking from texts. It's our happy college student there. How we read. What I'd like you to do while you're watching is take a moment to read the following sentence. Okay. Now count the F's in that sentence. Just do that one time. Don't go back and count them again. How many F's were you able to find? A lot of people point to, oh, this is going to be red on red. Let me switch my colors here. There we go. A lot of people point to this one and this one, and sometimes this third one over here. But there are actually 6 F's. We also have of, and of, and of. Here we can kind of see all of the ofs are in yellow and all of the other F's are in white. So I do this exercise just to kind of show you as like kind of an interesting way to think about the fact that a lot of times when we read, we don't always read that carefully. And just like missing the F's in this sentence, we sometimes miss major ideas or themes, or we don't really understand what's going on because we haven't done what's called deep reading. And often what I find is that people just kind of do skimming and they don't necessarily read. Or if they read, what they're trying to do for studying is read every single word the first time, and then they'll read it all over again for the test. Or they'll just read it for the test. So let's talk about what we can do instead. So the types of reading are skimming. That's where we get the surface ideas if we don't have time to read. And it's not a bad reading strategy in certain situations. So let's say that your instructor has assigned you a chapter, but they say also this other chapter would be helpful in understanding these concepts. Well, you might have to read the thing that they gave you a little bit more deeply, but the optional reading you could just skim through, and we'll talk about how to do that more effectively. Another type of time that skimming works is if you have procrastinated. I'm not saying that you should, but if you have, there are some tips and tricks to kind of help you if you get yourself into that kind of situation. And skimming can also be very useful when we're studying. So some things you can do here are reading quickly, focus on the headings and subheadings, anything that's in bold to cover the content at a general level. If this is a longer chapter, things like reading the opening paragraph, reading the closing paragraph, reading the questions at the end. Active reading is a little bit deeper. Active reading is where we read to understand and remember the information. So if you have a textbook that you are trying to memorize certain points from for an exam or for something that will apply later in a career, if you are reading a book for fun and you want to kind of understand what's going on in the book and remember the plot points. So here we focus on identifying main ideas and supporting points. You'll notice that this goes a little bit beyond just the overall idea, right? When we get with skimming, we get a little bit deeper in here. So identifying the main ideas and supporting points and memorizing them would basically be active reading. Deep reading goes beyond memorization. It's where you actually think about and analyze what you're reading. You compare it to other things that you've read or heard about. You criticize the author's ideas. You do further research, break their ideas open, and dig underneath the surface. And this particular type of reading, we encourage in kids, and we kind of let go of that for a lot of times for adults. But for adults who are in college, and even if you're 18 or 17, technically, if you're in college, we consider you adult in your learning, right? If you're an adult who's in college, the reason that this is important is because this is how adults learn. Adults learn by thinking about things, how they connect to their past experiences, how they're going to apply to their future experiences, why these things are relevant. And the idea of experience and your prior knowledge is quite important. So deep reading, if you're able to do it for your college classes or even on your own, helps you make those connections and not just memorize it for a test, right? Not just skim over it and kind of understand general idea of what's going on, but really get deeper into it and begin to apply it to your life. And that really is the point of college. A lot of people who say that college is pointless, it's because they're used to doing steps 1 and 2. They want to memorize things and pass the test and get a piece of paper. What I'm trying to teach you guys how to do is to go beyond that so that you can actually apply what you're learning to your daily life, which is, to me, more important. Deep reading for success. So, this is one method of deep reading. You don't have to do this formally. I think when I teach this, a lot of people think, oh, I have to make a lot of the Q stands for questions. So for example, somebody will say, do I have to write 20 questions before I read something? Not necessarily. It's more of a way of thinking about your reading, particularly when you have multiple classes and you're trying to not just skim the material, but understand and then apply it. So it stands for survey, question, read, recite, and review. So those are the 3 R's there. I mentioned that because last semester somebody asked me what the number 3 stood for. I was like, nothing. There's just 3 R's. All right, so survey. Survey is another term for skim, which I'm going to write in right there. So instead of waiting to the last minute and just skimming through, the idea here is that you're actually going to skim the material first. These are people surveying land, right? They survey things to see what's there. What do things look like? What are we going to find? So this happens. Oh dear, sorry guys. This happens before you read the chapter. There we go. So, read titles, headings, and subheadings, and I'll show you what that looks like. Okay, so this is an open source textbook online. OER stands for Open Educational Resources. It's basically a textbook that is free to the public. So, let's say that my instructor has assigned me chapter 2. That's on psychological research. So I would say the chapter title right here is psychological research. The headings for the chapter, why is research important, approaches, analyzing findings, ethics, and then they have key terms and a summary of the whole chapter. So that would be my first thing with skimming. I'd look over those titles. Okay. Looking at this, I know that this chapter is going to be about psychological research, why it's important, how people do it, and how they analyze their findings, and then this thing about ethics, right? How does ethics play a role in psychological research? So let's click. I have not seen this yet because I kind of wanted to look at it as someone would look at it. Okay, So our chapter title is Psychological Research. Our heading is Why is Research Important? And then this in bold is a subheading. Learning objectives. What they want us to learn in this chapter. By the end of this section, you will be able to explain how scientific research addresses questions, discuss how scientific research guides public policy, and appreciate how scientific research can be important in making personal decisions. All right, so just skimming that little bullet point list, I know what the section is going to be about, and I know what to expect, and I know what they want me to learn. And a lot of textbooks are set up like this. They'll introduce the material, they'll give you the material, and then they will review it, like here in this summary, right? So here are my subheadings. Use of research information, notable researchers, process of scientific research, and then some graphs and pictures and things of that nature. Okay, so I have just, in about 15 seconds, I would have been probably quicker heading up and talking to you guys, but in about 15 seconds, I've gotten an overview of this section. So let's look at what else we need to do when we're skimming that might take a little bit longer. Okay, so our first step is to read titles, headings, and subheadings. Our second step is to read the introduction, the summary, or the about the author section. So if you have a shorter article, a lot of times there will be a paragraph at the top or the bottom about the author. So here it's telling me to read the introduction, which I did with you guys, right? That quick bullet point list, and then the summary at the end, which we'll go back and do in a moment. Look at graphs and charts. There were a lot of pictures there and I skimmed through them, right? So any graphs, charts, photographs, images, anything like that you want to take a look at. Read any words in bold and read captions under the pictures. And then skim, again, the first and last paragraphs if you haven't done that already. So let's take a look back at our textbook. Okay, I'm going to skim over this part about the process of scientific research. So that's the subheading. I keep forgetting. Sorry, guys. That's the subheading. And I do have two terms in bold, deductive reasoning and inductive reasoning. Now, let's say that I kind of have an idea of what that means, but I'm not quite sure. So I know that I'm going to have to look up those words. They're going to be here in the key terms, so that's going to be helpful for me. Hypothesis or general premise, deductive reasoning, empirical observations, inductive reasoning, and then it kind of goes in a circle. And the figure says psychological research relies on both inductive and deductive reasoning. Well, that's pretty helpful actually to understanding that concept. And I kind of know about the scientific method already. So that's where I would connect my past learning. I think I learned this about the scientific method probably in a biology class in maybe 10th grade. But it's kind of the same idea. You come up with a hypothesis, you test the hypothesis, you observe what happens, and then you see whether your hypothesis was proven. And it looks to me like this is here. So that's what I mean by deep reading. I'm making that kind of connection as I'm skimming. I'm not just kind of going through and being like, okay, what does it address? Oh, there's the use of research information. Here's some people who are notable. Although I will say this, in a test, this might be just as an example. Let's say that this has a lot of information. They seem to just kind of be listing people here as I'm skimming it. I might not have to read that part that intently. I might not have to quite remember that if they're just listing people, unless my instructor goes into more things. So other bold terms, theory, hypothesis. Yep, that's what we were just talking about. And then we see again the scientific method. Have a theory, create a hypothesis, collect data, analyze it. Yeah, so it looks like this class, I do actually know some stuff here that could connect to my prior experience, and there's some stuff probably that I'm going to have to learn as well. Whoops. I don't know. My other thing started up. Okay, so then you would go here look over those key terms again, especially if there's some that you don't know. Maybe you haven't heard of confirmation bias before. So we'd kind of skim through this, right, as we're doing the S, dependent variable, double-blind study, empirical. Some of these terms might be familiar to you, some not. If they're not, you can kind of make a note to come back to them later or know that you're going to learn about them when you actually read. Then I could look at the summary. And again, if you were in a crunch, just reading these couple paragraphs would give you the overview of the major ideas in the text. So you could read that summary, and then you could be done with the first part of our method. So the second part of the SQ3R is the Q. And this is where we question before we read the chapter. This is the part 2. A lot of people think it has to be done formally. It does not. You'll notice I really did the question part kind of while I was reading. So ask, what did my instructor say about this chapter? If they said, hey, Megan, I want you to read chapter 2 and especially pay attention to the ethics section and also the section about how to form a research hypothesis. They might tell you things like that, what they're looking for, or what might be on the test. So that is important to know. And then I kind of asked as I was going along, what do I already know about this subject? So I know from high school classes about the scientific method. I don't really know how the scientific method applies to psychology. Or maybe I do know, but I don't really know what ethics might be involved. So that's something I'd have to pay attention to. And it can just be as quick as that. How does this connect to my past? How might I use this in the future? I'm not maybe going to be an education or a psychological researcher, but maybe I want to do something else where ethics are involved. Maybe I'm in the criminal justice program, and so ethics are going to be really important. What can I learn that I can apply to my life later, or at least talk about at parties and impress people? Develop questions you think might be on the test. You can do that too, a little bit more formally. That I typically, if I'm studying for something, and I usually am taking some sort of class, a lot of times for teaching. But if you are doing that, you can, while you're taking notes, develop questions you think might be on a test. You can turn the title and headings into questions as you're skimming through them. So, psychological ethics, what does that mean? Why is that important? Read the questions, the beginning and end of the chapters. A lot of times you will have professors who like to save time by using the questions from the book on quizzes and tests. So if you do that, chances are that you'll have a better chance of passing. I'll put it that way. All right. So we've skimmed through it. We've asked ourselves questions. What's next? Now we come to reading. I'm sorry, I don't know what that slide was. So now we come to reading. As you study, note all the underlined, italicized, or bold printed words. Study graphics, pictures, and charts as you're reading. Read the difficult passages more slowly. Read them twice if you have to. If you're confused about something, make a note about that. Read one section at a time. Now, this says recite after each section. It doesn't mean that you're repeating it back, but we're going to talk about what that means in a moment. Basically, it means that you are breaking things up into chunks, and this idea of chunking is quite important because our brains can only take in so much information at a time. So what I find a lot of times is that people try to cram for tests. They didn't read the chapters as they went along, or if they did, they didn't take very good notes, and they kind of try to read it all over again. When my brother was going through school, I talked with him actually about this method for better study habits, because that's kind of one of the things that he was doing. And then he'd be like, my brain is on fire. He's in a medical program, he just finished. So that's not going to work, especially if you have a good number of courses. When I was in college, I learned how to do this a little bit better because I was taking 21 credits all at the same time. So it would have been nearly impossible for me to just read without breaking things up apart. So look for important terms that you don't know or you need to define. And we're going to talk later about study habits for reading and how those can help you. So the other R is recite. There's somebody reciting. This is during and after the reading. I cannot tell you this enough times. Don't just highlight. You should highlight or underline, but don't highlight too much and don't over highlight. Sometimes I look at my students' books and they're like a whole page full of pink or a whole page full of yellow. That's not going to be helpful because then everything's in color and the main ideas don't stand out. So try to summarize what you've read instead. This is where basically at the end of a section, you write kind of your own notes. And we'll talk about different ways to do that in a moment. But for me, I write, sometimes I'll do kind of a little outline of what I just read. And other times I'll write a couple sentences, like kind of a short paragraph in my own words of what I think are the main ideas that I'll have to remember. Visual learners can picture the situations or concepts described. A lot of times visual learners like bullet point lists because they can see those bullets. They also like highlighting. Auditory learners could read their notes out loud. So this is something that I sometimes do as well. I do teach English, but I'm also an auditory learner. So I learn by hearing, and by hearing myself think about and analyze and summarize what I've read and how I'm going to apply it. Kinesthetic learners, this is my brother. Kinesthetic learners learn by movement. So visual is sight, auditory is sound, kinesthetic is by movement. So a lot of times people who learn by movement also learn by practice. So if you can find a practice quiz, if you can make up questions for yourself during the recitation part to quiz yourself. My brother also used to study by walking around. For him, moving and reading over his notes was helpful. So that's another thing that you can think about. And reader, writer learners sometimes can turn their list into an outline, again, similar to visual learners. So our next R is review. This is our last R. Review after you've finished reading. Read over the notes you took. Look over the text that you underlined or highlighted. Look over your answers to the questions at the end of the reading. Make flashcards for the questions at the end of the reading that gave you difficulty. And then you can, again, make questions to quiz yourself. So when you're reviewing, you can also go back and skim again and then read over anything that you forgot. I forgot to put that in here. But yeah, going back and doing the skimming part, again, can be quite helpful. So you will notice that what I do not have in here is read the entire chapter. Again, that's big waste of time. And if you have like psychology that we were just looking at as a good example, a lot of times the psychology class will have like a midterm where the first 10 chapters of the book are covered and a final where chapters 11 through 20 maybe are covered. You're not going to be able to read or reread all 10 chapters 2 hours before the test. You're just going to forget major things and you're going to get burned out and stressed out. So instead what you want to do is take notes while you read, which again we'll talk about, and skim through again to find those major ideas, reread the summaries, reread the questions, and then you can quiz yourself, and then you're reinforcing what you read before instead of just reading it all over again. Okay, we're actually just going to do just this one activity, and I'm a little bit longer than I wanted to be for this presentation. So the part about note-taking and studying, I'm going to do next time, and that video will be in Blackboard right along with this one. If you're not in my class, you can look for this under my name in the list for college composition. So this is Elba, and we're going to read a very short article about her. Yes, she is a green glow-in-the-dark bunny, and yes, she is real. So first, we're going to survey the article. We're going to look at the title, the caption, and any bolded words. Next, we're going to ask ourselves what we think the article might be about, what kind of class we might read this for. And then we'll read, and here I'll highlight a couple important facts if I'm able to. And then we will talk about what a teacher might quiz us on if this were part of a larger chapter in a book. So, title, A Green Bunny. Fluorescent, albino, pigment, illuminated, excitation, emission, synthetic mutation, gene, fluorescence, and mammalian. Okay. I want to go back. There we go. So we just skimmed through really quickly, and as I'm remembering it, the title is A Green Bunny, which is at least interesting. We have some scientific type of words, mutation. There are a few things that I know, like emission, but I'm not sure about some of the other ones. Mammalian means being a mammal. So those are questions that I'm asking myself. How scientific is this article going to be, and am I going to understand the science? That would be my question as someone who has not taken a science class in a very long time. Okay, so now we've done the S, we've done the Q. And also my other question is, why do we have a green bunny, and how did she become green? Alba, the fluorescent green bunny, is an albino rabbit. This means that since she has no skin pigment, under ordinary environmental conditions, she is completely white with pink eyes. Alba's not green all the time. She only glows when illuminated with the correct light. Okay, so I might, in my notes, I might put a highlight here, and I might underline this. Illuminated with correct light. When and only when illuminated with a blue light, maximum excitation at 488 nm. Now in my notes, I would put a little question mark there because I don't know what that means. Same thing here. What? I'm guessing by context that it has something to do with the amount of light that's emitted. I don't know what maximum excitation means in that context. She glows with a bright green light, maximum emission at 509 nm. She was created with EGFP. What the heck is that? An enhanced version, a synthetic mutation, of the original wild-type green fluorescent gene found in jellyfish Aquaria victoria. EGFP gives about a greater magnitude of fluorescence in mammalian cells than the original jellyfish gene. Okay, so my understanding of this, even though I don't understand every single word or concept here because they don't explain what EGFP stands for, those exact words. So to answer my questions and things that I would write down to study, the article was about a green bunny, a white albino bunny who glows in green light when she is illuminated with certain types of blue or of blue lights. And she was created with a gene from jellyfish that makes the jellyfish glow and can actually make her glow as well. I have a lot of other questions about this. This is something that an artist did, a genetic artist. That's what he considers himself. That I happen to know from when I found the article initially. But at any rate, this might be in a class for biology about ways that genes can be manipulated. So perhaps this would be a good example of genetic mutations or an example of genetic modifications. Maybe I would connect that to the idea of genetically modified food. This to me sounds kind of cruel to do to the animal, but I don't know that that's the case. But that would be a question that I would want to know if I were going to read more about it. So that's basically how this works. You can see we skimmed through, got the general idea of what was going on. Then we asked ourselves questions. What do we know already? How does it connect to what we're going to be doing in class or what we're going to do in the real world? Then we read and we made a few highlights, a few question marks that maybe I would ask a teacher later on what these things exactly meant, or maybe I would look up. And then the recitation would be making a little bit of a summary. And then if I were going to review, I would go back and probably, because it's shorter, I don't need to reread this, but I could read my 2-sentence summary about it. There was a bunny who was injected with jellyfish genes and DNA so that she glowed in the dark. When she was illuminated by a light, she glowed green. It's crazy, and it's an example of genetic mutations and gene manipulation. That would be my recitation. So I hope you guys can kind of see how this works, this method of reading. The next time we come back, we are going to talk about other important points of college reading, and then we'll get into study habits and some note-taking skills. Thanks, everybody. I'll see you then.